This is a great opportunity, wonderful meeting, and a great introduction. This is a great thing for me, so thank you very much. I recognize that I am the last thing standing between you and your lunch, so I will go quickly, I hope. But I want to, rather than a typical introduction, I want to just make some statements that are in and of themselves conclusions that will be topics for another day. We could probably spend half an hour on each one of these statements that I'm going to make, perhaps the poster session or afterwards. But what I want to get to is a recent story that has generated a great deal of interest. In the last 3 months since it was published, we've received multiple highlights by editorials already about this. And this is largely the work of Dr. John Clark, who is finishing up his postdoc now. And so the slides and the animations are really nice because he's going to go take it out on the roads, and so they're quite lovely. But it started off as a rather interesting story that began with Jay Gandolfi's retirement and the lectures that I needed to fill in my drug metabolism course. And so I asked a colleague, could you pick up a couple of lectures? And he said, well, sure, I could do pharmacogenetics, but I would like to take 5 lectures minimum. Well, sure, I'll love to let you take 5 lectures, but you realize that it's not just genetics that's the determining factor. And so we started this playful banter back and forth about whose dad is stronger than whose, you know, rather degenerated into a juvenile discussion. But really, it started a thought process on my part that, you know, how would you test that? How do you test? Genetics is far and away advanced in, technologically. The amount of money that's been spent on it, the energy that has been put into genetic research on inter-individual variability and drug response, and certainly public perception. They're light years ahead of us. But they've also recognized that genetics isn't the answer for everything. And so the question of how can we determine whether the major driver of inter-individual variability is genetics or disease or some combination in between is really what I want to get to. And so there's a couple of statements I'm going to make. The first is the prevalence of steatohepatitis. There's a whole lot of people with NASH. We all know that. But what I want to make the point here is that the number of patients who have been actually diagnosed is very small. So you ask yourself, why isn't NASH already shown to be a major factor that correlates with adverse drug reactions? Well, if you thought that you could predict a pattern and you made your first pick and 99% of the time they lied to you, you're never going to be able to predict that. And so if they simply don't know that they have NASH, the only way to diagnose it is with a needle biopsy. And if it's not known which patients have this disease, how can it be predicted? How can it correlate? So you'd have to predict, well, are there other factors that also correlate with NASH, such as obesity? And there are certain cases where adverse drug reactions correlate with things that also correlate with NASH. The second one, does NASH itself create change in the individual? So we did actually Dr. Lake, who's here, did a study with human samples at the various stages of fatty liver disease and found that rather than sex or age, the major driver, this is a principal components analysis, the major drivers of gene expression variability, and what they found was that age and sex weren't really all that discretionary, but what the main driver was disease status. So the NASH humans had major changes that grouped together, and they were the major drivers in the principal components analysis. But that's great for general assumptions. But every drug that we take and every adverse drug reaction has a story. And that story has a molecular mechanism. And so you have to look at the individual elements of pharmacokinetics and the fate of each drug and make a story. And so our laboratory has spent a great deal of effort to try to elucidate some of those mechanisms and make stories. And so the introductory talks that we've had today are excellent. Phase 1, metabolism, 2, conjugation, and then disposition through uptake and efflux transporters at the different sides of the cell. So we have shown multiple mechanisms by which regulation of this gene expression and function happens. So this is function of some of the major P450s, a few of the major P450s. What you'll notice is here, there. So 1A2 metabolism of phenacetin is actually downregulated in the disease, and that follows transcription. So mRNA and protein and function are all downregulated, so the typical transcriptional regulation. But hydroxytolutamide and diclofenac metabolism are both metabolized by CYP2C9. And so 2C9 is downregulated post-translationally because there is no change in protein levels. So despite no change, there's a clear decrease in function of 2C9. We've also seen some odd cellular localization events. So MRP2 is one of these transporters. It has to reside on the canalicular membrane in order to take substrates from inside the hepatocyte and put them into bile across the canalicular membrane. And if during, and it has been shown in vitro, and now we've shown it in vivo, that in periods of stress, like steatohepatitis, MRP2 actually moves away. It's a cheap way for the cell to regulate function without having to degrade the protein and then make it in a de novo synthesis. And so it's a quick and easy way for the cell to regulate function by moving it away from the membrane. And so you'll see here that this vesicularization event, there's really a canaliculus there, but the MRP protein has been moved away so that it functions less. And that has resulted in changes in disposition. Here we look at acetaminophen disposition, and one of the substrates of that transporter is acetaminophen glucuronide. And so in the animals that have NASH, the glucuronide substrate of MRP2 is retained in the plasma because it doesn't go into the bile. And that is also replicated in humans. This is a pediatric study where we have seen that children with NASH actually have altered disposition so that the metabolites, the MRP2 substrates, are retained in the plasma. Well, there are many drugs, and acetaminophen glucuronide isn't probably the best substrate, but there are other substrates that are even better substrates for MRP2, and their disposition is even altered more. And what that means then is that toxic substrates for these transporters, if they are retained in the plasma, will display not just hepatotoxicity, but multi-organ toxicity. We've shown toxicity in multiple organs because of plasma retention. Here, this is methotrexate, which is also a substrate for MRP2. We have healthy livers. We have the disease pathology. Then we have a drug-induced toxicity in the healthy livers, and then a drug-induced toxicity in the disease. And because there's more drug that's retained in the plasma, we see hyperplasia, we see greater liver toxicity, as well as renal toxicity and others. But also, importantly, is that because they're staying in the plasma more, they're not going back to the bile. So here we have fecal elimination. The control healthy livers are putting the methotrexate into the intestines for fecal elimination where it belongs. Importantly, for patients receiving methotrexate, leukemia patients, the first indication that they've had enough is that they have toxicity, they have diarrhea. And so without that diarrhea, these patients would have, so here's supposed to be drug-induced toxicity, but because the drug is staying in the systemic blood, the toxicity is in the healthy animals, and so that might have clinical implications. That's a different story. And the last one that I want to make is that the intrinsic versus the idiosyncratic toxicities. Most of the toxicities that we know of are dose-related, are dose-dependent. And even when we can't determine a mechanism, we classify them perhaps as idiosyncratic, when in fact they are more sort of an inter-individual variability of drugs that really classify them into the dose-related intrinsic type of of adverse drug reactions. And so the idea of altering drug metabolism disposition so that you have systemic exposure rather than typical hepatic clearance, if you have systemic retention, then you're going to predispose those individuals to greater toxicity, multi-organ toxicity. So now with that sort of background, the story begins with what would be a fair playing ground for our genetics versus environment sort of test. So it has to be fair. You can't stack the deck one way or the other, even though, you know, of course I'm right, but no, I'm just joking. So you have to have a legitimate genetic story, but you also have to make it a legitimate environmental impact. And so Dr. Slip mentioned the great example. So the statin drugs are transported and eliminated, and in certain individuals there is a risk of myopathy. And so they did a genome-wide association study. This was one of the foundational. It's a famous genome-wide association study, and it identified one gene, one polymorphism in an OATP1B1 transporter. It takes statin from the blood and pulls it into the liver for elimination. And that polymorphism makes it so that those patients can't pull it in. And so the toxicity with the homozygous polymorphic patients versus the reference allele, they have a much greater incidence of toxicity over time. So that's the genetic component. That's a fair genetic story. It's a famous genetic story. But on the environmental side, we have shown that, so we took the human progression of all genetic variability, and we have, so the green parts are so on this bar here, this is a comparison of healthy livers versus NASH. And so there are a handful of genes, about 10% of genes are upregulated by NASH, and about 10% of genes are downregulated by NASH. And so if you took a subset of the 32,000 genes in our bodies, if you took some random subset, you would still expect to have about 10% upregulated and about 10% downregulated if, of course, it was a random subset. And so when you take all of the drug metabolizing enzymes and you do an analysis there, there's, instead of 32,000, there's 250 some odd. And so there's roughly 10% that are upregulated and roughly 10% that are downregulated. And so drug metabolizing enzymes aren't specifically targeted, but remember, it's all individual, it's all mechanistic based. And so if one drug metabolizing enzyme is changed, then that matters. But when we did an analysis of transporters and even broke it down into just uptake transporters, we found that the number of genes that were downregulated were significantly overexpressed. And so that is this analysis down here. The actual number of genes that we found that were downregulated in NASH was much greater than what we expected through a normal distribution curve. So it should have been in this gray bar area, but it was significantly higher, suggesting that transporters, uptake transporters as a whole, are specifically targeted for downregulation in hepatic cells that are stressed. They don't want to take more in. So I think that this is a fair fight. So we've got a genetic component, we've got an environmental possibility. I'm rooting for one side, of course. Okay, so the natural story of statins. So there are multiple uptake transporters. So I remembered a paper that was written by Richard Kim several years ago about statins and how there were multiple transporters. Even though there's that one major transporter, there are multiple uptake transporters with overlapping substrate specificity that can compensate for loss of one of the transporters. And so in a case of polymorphism in one transporter, you've still got compensatory transport. And so there are at least four transporters that take up the statins, and there are some backwards and some canalicular transporters involved, but this is an uptake story. So statin will enter in the blood, And there are some minor players, NTCP and OATP2B1. And then there are also some major transporters that result in uptake from the plasma. Then there are transporters that put it into the bile and for elimination. However, so there we have minimal risk of adverse drug reactions and no real increase in plasma AUC, which is a measure of exposure. However, in the polymorphism, there is a loss of function, a significant loss of function. It's not a complete loss of function. It's in like the teens, but it's a significant loss of function so that when that major transporter doesn't participate, you've still got the compensatory minor players, one of the major players, but because of that loss of function, the statins will stay in the blood. You'll have a modest increase in plasma concentration and exposure to the drug, and a moderate increase in risk. So here's what that data looks like in the humans. So the middle bar is the reference allele, and the higher bar is the polymorphic patients. And so what you'll see then is a modest increase in plasma exposure in these individuals that, again, leads to an increase in risk. And then for simvastatin, again, simvastatin is not a substrate. It's the simvastatin acid. It's a metabolite. So half of the statins are metabolized and half of them are not. And so here, this is the substrate, and so it's the simvastatin acid that in the polymorphic patients that has a much greater exposure. And so to make it a simplified story, we stuck with pravastatin, and we used the associated transporter that correlates to the human polymorphism, and so we had a knockout of OATP1B2. And we gave both the wild type and the knockout animal NASH. And so this is an indication that, yes, indeed, they got NASH. All of the indications of NASH, so all of the NASH animals got NASH, whether wild type or knockout. And so then our first interest, of course, was in the transporter expression that drives the exposure and elimination. So the first one was the OATP1B2. We were hoping it was a knockout, and indeed, it is a knockout. And so that's nice. But then in the wild-type animals, the result of the disease was a decrease in protein expression of that major transporter. And other compensatory transporters, we see the same pattern, where we have a decrease in the disease for multiple compensatory uptake transporters. However, there was one that was kind of odd, kind of threw me off. Even though several of the uptake transporters are downregulated, not all of them are. And so 1A4, one of the other compensatory transporters, was actually upregulated in NASH. So when we found out the results of this study, I was actually quite disappointed. So NASH, in and of itself, alone, only had very minor increase. I was disappointed. I was cheering the wrong way. But when we looked at the knockout animals, it was exactly the way it was supposed to be. It had been reported before, and then we repeated that, and that there was a modest increase in plasma retention. And this is a log scale for pharmacokinetic analysis, and so this is, this is a moderate increase in plasma AUC. However, when we took both the knockout with the disease, we showed a significant increase, a synergistic increase. And so when you look at exposure, plasma area under the curve, you see the moderate increase in just the knockout alone. But then when you put knockouts with the disease, you get a 10 times greater plasma area under the curve, significantly increasing their exposure to the drug. Now, because this is a muscle toxicity, it's a dose-related muscle event, we also looked at muscle concentrations of the drug. And in the gastrocnemius and soleus muscles, we see this significant increase in exposure inside the muscle. So to demonstrate what we think is going on, in humans with a with the knockout, we have a loss of function of the major transporter and a downregulation of the compensatory transporters. So this is in the animal, by the way. And so what we see then is the other transporter can still transport, but because of the downregulation of two of the compensatory transporters and the loss of the one major transporter, we get a significant increase in plasma AUC and increase in risk for adverse drug event, which is nice if you want to keep your pet rat healthy. But what's happening in the human? So the major transporter, OATP1A4, the homolog in the humans, is actually elevated in the disease. So the protein levels are increased in human NASH. And the compensatory transporters are downregulated in human NASH. And so what we think would happen in this study, if we were lucky enough to have a whole bunch of biopsy-verified NASH patients who are also genetically screened for the polymorphism, which we calculated we would have to take everybody west of the Mississippi for 5 years. So that's not going to work. So in humans, because of the polymorphism, there's a decrease in function of the major transporter. And if those patients also were to develop NASH, even though there's an increase in expression, the loss of function is due to the polymorphism. So there's going to be greatly reduced function of the major transporter, and the downregulation of the compensatory transporters should result in a much greater plasma area under the curve, exposure, and risk of toxicity. So although my team didn't win, we demonstrated that environmental factors such as NASH play a major role in not just OATP uptake transporters, but we've demonstrated multiple molecular events that have happened that happen in response to NASH that predispose patients to adverse toxicity, and that if our efforts to provide precision health to patients, if we're going to do that effectively, we have to account for things that drive those types of changes, like NASH. I have many people to thank, particularly John Clark and April Lake and Annika Jerlenga. Happy to talk to anyone about these. We've got 2 posters, and happy to talk to anyone. Thank you very much.
Thank you, Nathan. Let me start with a quick question, Nathan. The fact that you didn't see any simvastatin AUC changes in the NASH animals, not the knockouts, but just NASH only, did you look at kidneys to see whether some kidney transporters might be compensating for? Yes,
so the kidney is a new effort of ours with its own problems. We have shown that there are some. We haven't published it yet. I think we've submitted it to whoever gets that one. But we've seen changes in renal transport expression. But for us that brings up a whole other problem, because if we're going to correlate the changes in renal transporter expression of patients with the liver disease, we can't really use, animal models. There's no really great animal model. And so what we did is we used 7 different animal models. We compared the transporter expression in the animal models versus humans, and we showed which ones are the best for pharmacokinetic analysis in the animal models. But then we started to look at what happens in the kidney. And because the biochemical events are so different for these models, what we really need, but is not available, is a list, is a biorepository that has kidneys that are cross-referenced to patients who have had liver disease. And so you can't just go up to the drive-through window and say, I'd like kidneys from patients with liver disease. They don't keep that type of information. So one of my graduate students, actually, he's from Georgia, and his brother, serendipitously, is the pathologist in our division of, he catalogs, he's the cataloger. So we're going to have to take him out to dinner a lot and try to figure what we can do to see whether there is renal compensatory changes. NASH is not the only way that it's well known that the kidney compensates in multiple types of stress, like cholestasis. The kidney can compensate for hepatic dysfunction. And so that's naturally an interest of ours in the future.
Maybe time for one more question, perhaps? We're running later.
Or it's lunchtime.
Something that's missing from a lot of the discussion here today is self-proliferation. I brought that up at least once, but I mean specifically, we did a study in OB mice, which are prone to the increase of liver cancer at about 1 year. We looked at several potential risk parameters, and the only thing that was changed significantly was increased self-proliferation. And I've yet to see a paper here that shows this for humans. It's just not being discussed.
I have some data in my computer that I can show you. I'd be happy to sit down with you.
Great discussion for the roundtable. Thank you very much. Thank you.